Welcome back, friends. Welcome back in a new video lecture series where we are going to talk about recombinant DNA technology. So it will going to be a very vast video lecture series where I will provide everything about recombinant DNA technology that one need to know. Okay, so today's video is just an introductory video where we are only going to give a big picture of recombinant DNA technology because it is a subject that if you first clear the big picture, then everything becomes easy. So we will first do this. Okay, so what recombinant DNA technology is? Recombinant DNA technology is simply the process by which we can clone our DNA molecule, okay, with the help of the vector molecule and the host. That is the idea. So recombinant DNA technology is simply the process of DNA cloning. So what DNA cloning is? So first, DNA cloning is the process by which we will have multiple copies of identical DNA molecule from a single identical DNA molecule. What it is? Suppose you have a mobile phone and you need to prepare multiple copies of same mobile phone with the help of this single one, and this is simply what DNA cloning is. With the help of a single ancestor DNA molecule, we will have multiple copies of identical DNA molecule. That is the overall idea. So we are going to see what DNA cloning is in a very brief manner. That is what our introductory video. So in DNA cloning, we will have the two process by which we can perform our DNA cloning. One is cell based DNA cloning, in which we will undergo the whole process inside a cell, and another one is cell free DNA cloning, that is our polymerized chain reaction. We will not going to talk about this here, we will talk about in some other videos. Okay, so in cell based DNA cloning, we are going to see what is going on in a very brief manner. So suppose this is our human chromosome, where we are going to find the target sequence which we need to amplify. So suppose this is our target DNA sequence. Suppose this is the sequence which can ultimately code a gene, and that ultimately give us a protein. Suppose this is a gene which can prepare insulin protein. Okay, so what we need to do, first of all, from this bigger chromosome we need to cut it. So we need something called molecular scissor. Okay, as we need to cut the whole the target sequence from this whole chromosome. So what will be our molecular scissor? Molecular scissor will be something something called restriction endonuclease, and it is a type of enzyme. Okay, and this restriction endonuclease will be specific for both the chromosome and the vector by which we will transfer our target DNA sequence into a host in which we are willing to amplify the target chromosome. Okay, so here we take the restriction endonuclease A. This is a hypothetical term. Example of restriction endonuclease in reality are something like BAM H1, eco R1, just like that, and we will also talk about this in our later video, so you don't need to bother about this, just understand the whole process. So we have our vector. Suppose it is a vehicle by which our target DNA will enter into the host chromosome. Okay, so host cell. So we will cut both a specific portion of vector and specific portion of target DNA with the help of restriction endonuclease. So restriction endonuclease will cut specific sequence of vector and specific sequence of our chromosome, and then we have our cut out portion. Now we need to fix this in the vector chromosome. Okay, so we will do this with the help of an enzyme called ligase. So ligase will be referred as molecular glue. Restriction endonuclease referred as molecular scissor, as they will cut the target DNA sequence and the specific sequence in which the target DNA sequence will attached in the vector. Okay, so they are referred to as molecular scissor, and ligase will attach the target DNA in the vector, so we can call it molecular glue. Now this vector we can also call this replicon, as they have the ability to independently replicate. So what is replicon is? Replicon is simply something that have the ability to independently replicate without help of any host chromosome. Okay, so after target DNA ligation with the help of the DNA ligase, we have our recombinant, and this vector will now refer to as recombinant, or we can say it is chimera. And that is the reason why the process name is recombinant DNA technology, because we are going to take the help of recombinant to produce our target DNA sequence in multiple. Okay, so chimera or hybrid or recombinant consist of autonomously replicating DNA segment and an inserted segment. So what chimera is? Chimera or hybrid or recombinant is simply consisting of a autonomous replicate DNA molecule and inserted segment. Inserted segment is our target DNA sequence, which we need to convert into multiple copies inside a host of our desired. Okay, so suppose here the vector is plasmid. Okay, so if it is a plasmid, so and we need to insert this in a bacterial host, so what we need to do, as we all know, to increase the competence of bacteria, we need to add calcium chloride. If we add calcium chloride here, they become so crazy for taking the plasmid or another extracellular DNA molecule. Okay, so we will add our calcium chloride to make it more competence, and this process is called transformation, by which we help the recombinant or chimera to enter into the host cell of our interest, in which we need to multiply or amplify our desired DNA molecule. Okay, now there is a, we need to find out that whether the recombinant vector is entered into the host cell or not. So in that case we have three combination. In one case the host cell have the recombinant vector, in another case the host cell have only the non-recombinant vector, in another case there is only the host cell, that means transformation is failed. Okay, so we need to first of all select these two, that is whether vector is present or not. So we need to perform a selection of transform cell, where we will have a selection of recombinant vector and the transform cell with non-recombinant vector, and then we will again perform a selection process whereby we will select the transform cell with recombinant vector. Then we will have our recombinant vector containing transform cell, then we will amplify it, right? We amplify it and have our multiple copies of identical DNA molecule. Okay, now if we want only the recombinant DNA or multiple copies of identical DNA, then we will extract from the host cell in this stage, and if we want our desired product in protein manner, then we will allow them to undergo transcription and the translation, and then we will have our protein in multiple copies. In that way the overall process is going on. Now there are multiple questions remain. So what these vector are, what recombinant restriction endonuclease are, what ligase are, how this transformation process going on, I will go to tell you all about this in a very detailed manner in our upcoming videos. So this is the overall big picture of recombinant DNA technology, or you can say the DNA cloning, and this is the introductory video. In our future video we are going to talk about the different enzymes that are involved in recombinant DNA technology. So stay tuned. Thank you.